Okay. Welcome to those joining. We'll give it a few minutes to let some other folks roll in. We'll maybe get started in a minute. There's still some more folks joining. Welcome to those joining. We're just going to wait one more minute and then we'll start. Okay, well, lovely. There might be a few more joining, but I think we can get going now. So hello, everyone, and welcome back to the fall season of Ask an Expert webinar for 2025. My name is Meg Mills, and I'm the International Watershed Coordinator with the Lake of the Woods Water Sustainability Foundation. I also work part-time with the International Joint Commission, collaborating with partners across rainy Lake of the Woods Basin. I'll be your host for today's webinar, as well as the rest of the season's sessions. I'm also supported by the Executive Director of the Foundation, Tika Newton, Tika, who will assist with moderating the webinar during the Q&A discussion section. I'm joining you today from Treaty 1 territory. It's quite damp outside and a little cold, but another great day for learning something new. And today we're also joined by Dr. Chelsea Rochman, Assistant Professor in Ecology at the University of Toronto and co-founder of the U of T team to share insights from the pELAstic project she has led. Plastic pollution is one of the most widespread threats to freshwater, yet much remains unknown about how microplastics move through lakes and affect ecosystems. Chelsea's project, based in the IISD experimental lakes area, is leading groundbreaking experiments to track the fate, impacts, and recovery of lakes exposed to these microplastics. Dr. Rochman will share what her team is learning about how microplastics affect everything from plankton to fish and how lakes respond over time, and what this means for monitoring and possibly policy in watersheds like Lake of the Woods. We're really excited to have her with us today and unpack one of the most urgent and fascinating challenges facing our freshwater environments. Before we dive in, I just want to go over a few housekeeping points. This session is being recorded, and you'll be able to rewatch it later at our website, lowwsf.com/ask-and-expert, along with our past sessions. If you're new to Ask an Expert, welcome. This series highlights the incredible science and stewardship happening across the Rainy Lake of the Woods Basin and beyond. I assume most of you are familiar with Zoom at this point, but a reminder, I believe you're all muted right now, so we don't have to worry about any unnecessary sounds. Cameras are also off. At the end, if anyone has questions, you can raise your hand and I can manually unmute you so you can ask a question. At that time, you might also have to unmute yourself. I'm not sure about that. If you're using the mobile app, you can access Zoom controls by tapping anywhere on the active screen. A taskbar should pop up on the bottom of your screen where you can unmute yourself and shut off your video as well. You can also restore your sound and video on that tab. There should also be a Q&A box at the bottom where you can type your questions in there at any time throughout the presentation, and we'll keep an eye on those and save those for the end of the session. You can access this chat window through the bottom menu and type your questions there at any time. Everyone will see those chat comments unless you target them to a specific participant, but when we download the session, I'll be able to see those, so just keep that in mind. I'll also do my best to keep an eye on the chat and the raised hands throughout the session and reserve time after the presentations for questions. Oops. Just here's a visual of where everything is, the chat button, the raise your hand, and the Q&A. So again, today we're joined by one presenter, Dr. Chelsea Rochman, who is an assistant professor in ecology at the University of Toronto and the co-founder of the U of T Trash Team. Chelsea is also an advisor to Ocean Conservancy and research fellow at the IISD Experimental Lakes area. Chelsea has been working on the topic of plastic pollution for nearly 20 years and has won awards for her research. Chelsea also received her PhD in ecology from UC Davis and San Diego State University. In addition to her research, Chelsea routinely advises local federal and international government bodies on policies related to plastic pollution. So now let's get on with the webinar. Chelsea, I'll hand things off to you.
Thank you so much, Megan, and thanks, Tika, for inviting me. I'm going to see if I can share my screen.
There we go.
Can you see that okay?
Yes.
And you're seeing the slide and not my notes?
Correct, yeah.
Perfect. Okay. Well, thanks everyone for coming. What I'm going to do today is talk mostly about the plastic project, which is our experiment on microplastics happening right now at the Experimental Lakes area. But I also thought that I would give a bit of a background to myself and plastic pollution in general, and try to squeeze in some talk about solutions at the end, as long as we have time. I also want to make sure that we have time for questions. So first I'll start with just a little bit about me. First of all, I am a ecotoxicologist. I study pollution. I've always been really interested in how pollution impacts ecosystems. And I have a real passion for aquatic ecosystems, both marine and freshwater. And so I've spent all of my time working in aquatic ecosystems. And I also think this is relevant to pollution because the solution to pollution is often considered dilution, meaning we tend to think that by putting it into these large water bodies, they will get treated because they will become diluted in the system. But I have spent the bulk of my career researching plastic pollution. I've done that since about 2007 or 2008. So I'll start with why. And the reason is because it is right now a contaminant or a pollutant that's pretty much everywhere. And I'll talk about that in a little bit. But I'll start with how did we get here? And so this slide shows the cover of Life magazine just after World War II, during Industrial Revolution, where we're starting to create a lot of technologies that can make life for us more convenient, and plastics was one of them. This article here is celebrating the rise of, or the creation of, these single-use plastics that can make our lives easier. For example, having people over for dinner and simply throwing away the dishes at the end of the night, which of course sounds amazing, but this increase in convenience have led to unsustainable practices, which have led to a large pileup of plastic waste globally. And so here I'm showing you a paper led by Roland Geyer that was created in a working group that I was part of when I was in grad school. And basically that blue arrow shows all the plastic that was produced, 8,300 million metric tons, as of the year 2015. And then it shows of that amount, how much is still in use. So that's that 2,500 number. So about a quarter, a little more. How much has been recycled or incinerated, sometimes waste to energy, sometimes simply burned, which can put hazardous emissions into the environment. It's about 10% each. We are not recycling a lot of the plastic we are using. And over half of it, that 4,900, is discarded. And that's either into landfill or the environment. And landfills really range from heavily engineered to a dump, which will leak into the environment, to, of course, mismanaged waste. In this case, which is not a landfill, but in litter. So we don't quite know exactly how much enters the environment every year versus how much is put into different forms of landfill. But we have a pretty big understanding that the contamination of our plastic waste is global, that it's widespread in marine and aquatic ecosystems. And these images that you see are pictures that I took, or a colleague took, in the Great Pacific Garbage Patch in 2009. And sometimes we see these big items of plastic floating in the middle of the Pacific, far, far, far from land. But the majority of what you see here, this is an image snapped from the bow of the ship, are small pieces of plastic about the size of a pencil eraser and smaller. And these are what we call microplastic. And as an ecologist, these are concerning because they can get into every level of the food web and eventually lead to harm. So the majority of my talk today is going to focus on microplastics, which are a diverse contaminant, and I tend to call them a multidimensional contaminant in that they're made out of many different polymers. Then those plastic products have a lot of additives in them to make them colorful, flame retardant, flexible, durable. They get made into many, many different product types, which will have different sources or pathways to get into the environment. They break down into smaller and smaller pieces of different shapes. They can be fibers or they can be jagged fragments. They can also be film or foam. They range in color, which matters a little bit because sometimes animals choose items of a specific color to interact with. So that can affect exposure, which would affect risk. And then once they're in the environment, they accumulate some of those other contaminants that are in there, like PCBs, mercury, etc. So I like to think of these as these tiny little particles that range in size and shape and type and chemical load that can have impacts on animals and interact with animals. And over the course of my career, I'd say what we've really learned is that on the left is that microplastics are everywhere. They're in freshwater, they're in marine systems, they're circling in the atmosphere in the dust cycle, they're getting into soil, including agriculture, and they're moving through the water cycle as well as through food webs. So this really just means, okay, this is a contaminant that's really pervasive. It's really now interacting with a lot of biology, from cells up to animals to communities and populations. And so it's really important that we better understand it. So in my ecotoxicologist lens, I try to ask questions about how does this contaminant impact our ecosystem? And I like to do experiments in my career. I am an experimentalist, and I really see value of doing them at different scales. I see the value in doing laboratory experiments at very small scales with really high degrees of control to better understand exactly like the mechanism of impact. I see a lot of value in going out into the environment and making observations to get to understand kind of these natural experiments in nature to say, how are animals contaminated? Are they contaminated? By how much? Do they seem to be correlated with specific effects? But I see really great value in these large scale experiments that are a little less controlled, but that really allow us to understand how a complex contaminant interacts with a complex ecosystem. And so that's why I want to highlight the work today at the Experimental Lakes area, because you really are sitting, you're much closer right now than I am here talking to you from Toronto. But we really are sitting next to this almost one in a world, once in a globe type of experimental research station that allows us to manipulate whole lake ecosystems to really understand how stressors impact ecosystems to inform and guide policies to protect global ecosystems. And that's really what ELA does. So I'll talk about that. But before I talk about the plastics project, I'll spend a little bit of time talking about some of those experiments at different scales to show you a bit more about the issue of plastic pollution, both in the Great Lakes and also in the ocean. So I'll start with an experimental observation that we made in the field. This is Madeline Milne. She is a researcher in my laboratory. She did this work as an undergrad. She worked closely with the Ministry of the Environment, Paul Helm and Satunda Babsar, basically who were interested in how much microplastic is in our sport fish, because they try to recommend the amount of fish that people can eat relevant to health. Now, for microplastics, we don't have those thresholds and those amounts, but they wanted to get a handle on how much microplastic is in there to eventually see whether they should try to inform guidelines for safe consumption. So Maddie took several different fish from, this is Humber Bay in Lake Ontario, right by Toronto. For those of you that love sport fish or that love fish, they don't have to be for the sport of fishing. These are brown bullhead, largemouth bass, northern pike, rock bass, smallmouth bass, and white sucker. And here you can see the number of particles in the stomach of the fish. And this is the distribution in each species. And here you're looking at the number of particles in the fillet, the muscle of the fish, the part that we eat. And what's striking here is that we see microplastics in almost every fish. The amount in the gut can be over hundreds of pieces in an individual fish, sometimes 1,000, at least in this one white sucker. And in the fillet, we were really surprised to see pretty large amounts outside of the gut in the muscle. Again, the part that we consume, tens to sometimes hundreds of pieces. So this really begs the question, what are the impacts of this high exposure of microplastics to fish? So another student in my lab, Kennedy Bucci, who's recently done a PhD and now living in Thunder Bay, doing research at Lakehead University, she did an experiment to basically test how these microplastics would impact fish. And she chose to use fathead minnow, which are a really important prey fish, sometimes used as bait fish for a lot of the fish species that you see here. And so she did experiment where she raised fathead minnow fish in a laboratory. So this is a small scaled controlled experiment where she exposed the fish to lower or higher concentrations of microplastics that are environmentally relevant to the fish from their embryonic stage to adulthood, and then mated them and raised the offspring to say, how does the exposure of microplastics through their life cycle impact the fish themselves and their offspring, or the babies that they have? And the offspring were not exposed to microplastic. She exposed them to plastic that came from industry or from manufacturing, polyethylene particles that we were able to purchase off the shelf, as well as polyethylene that we picked up from the shores of Lake Ontario to have that chemical cocktail that was already in the lake. So I'll show you some results from that study. So remember, we had two different concentrations of microplastics, low and high. We had ones that were pre-consumer taken from the shelf and ones that were taken from the beach. We ground them into fragments of microplastics, and we tested a lot of different effects, but here what I'm going to show you is effects relevant to endocrine disruption or that population level effects based on reproduction. So here what I'm showing you are, so there's 5 tanks for every treatment. These are adult fish that started laying eggs on the experiment. And you can see that for the environmental microplastics in the lower concentration, those fish started laying eggs later. So they were developmentally depressed. The eggs that same treatment laid tend to be more friable or easy to break. And often those eggs led to dead or unfertilized offspring. When we looked in those adult fish, the fish in that same treatment had a much thinner chorion, or essentially like an eggshell, but in the gonad of a fish, not like a laid egg in a bird, but it was much thinner, which is likely what led to these more breakable eggs. And what we found to be most striking is that in the same treatment, the offspring, or the fry, the babies from these fish, had higher incidences of deformities than other fish in other treatments. So basically here what we saw is in the lower concentration of the microplastics taken right from the shores of Lake Ontario, we're seeing signs of endocrine disruption. And now you're probably thinking, why do you have more of an effect at the lower concentration than the higher? And this is actually pretty typical of endocrine disrupting compounds. And I can answer questions about that later if you'd like. We also tried to synthesize data from across lots of different studies to try to understand risk. We do a lot on risk assessment in my laboratory. Karen Kidd, Rebecca Rooney, and Eden Hatley will speak, I believe, next week about an IJC work group on microplastics that I co-led with them. They're going to talk about our risk assessments for microplastics. So I didn't want to show you that today, but what I'm showing you today is very similarly a risk assessment for macroplastics, which is a study led by Dr. Erin Murphy, which will be published in the Proceedings of the National Academy of Science probably next week. And here what we did is we looked at animals that were washing up on shorelines that had ingested plastic and had a necropsy to say, did this animal die and did it die from plastic? Synthesizing that to understand how many pieces of plastic does a seabird, a marine mammal, or a turtle have to eat to have, say, a 20% likelihood of death, a 60% likelihood of death, and in this case, what you're seeing here is a 95% likelihood of death. This can be used to try to inform risk assessments to reduce the amount of macroplastic bottles, fragments, bottle caps, lighters in the environment to try to protect, in this case, these large taxa. But most of the work that I do is on microplastics. So when I turn to talking about the experimental lakes area, I'm going to do a full focus right now on the small stuff, back to those microplastics. So many people in this room are likely familiar with the IISD experimental lakes area. I think Vince Pallas presented to you last week, and he is the head researcher at the station. But for those of you less aware, you can see here on a map where it is, right between Dryden and Kenora, or to give you a bigger spread between Winnipeg and Thunder Bay. It is a research station that was set aside by the government about 60 years ago. It has also about 60 lakes that were set aside for research, some that can be manipulated and some that are just used for long-term ecological monitoring to understand how systems naturally change over time with climate change, etc. Within these lakes, they have been used for a lot of different manipulative experiments, including looking at how aquaculture impacts lakes, to looking at chemicals like cadmium or mercury, and also looking at things like if you change the hydrology, how does it impact the lake? So these lakes can really be used for a lot of different questions. And a question I get asked a lot is, why are whole lake experiments necessary? How do you get permitting to literally essentially pollute a lake? And is this a good idea? And I understand those questions. And sometimes I think about them quite a bit when we're actually doing experiments, adding plastic to a lake. And I always try to remember to really thank the lake for what it's giving us. But I do think it's really hard to understand how contaminants are impacting a natural system when a natural system, for example, the Great Lakes or even Lake of the Woods, although Lake of the Woods is more remote, is impacted by many different stressors, be it overfishing, climate change, chemical pollution. And it's really hard to do those observational experiments in nature and tie what you're seeing to an impact. But policymakers want to understand the impact of pollution or the impact of a stressor in order to allow a change to be made. And so it is the best way to understand these complex questions. It bridges the gap between these smaller scale laboratory studies and even those big models to ecosystems in nature. And it is essential to contributing to policy, which ELA has done again and again and again. And for plastics, I've been able to really see how doing this research can also make a difference in the policy sphere. So for the pELAstic project, here you can see our funders. I'm putting all my acknowledgements throughout the talk instead of saving it through the end. But really what I want you to focus on this slide is what we're trying to use the lake to better understand, and that is the physical and the chemical and the biological impacts of microplastics, how they impact aquatic ecosystems across every level of biological organization, from cells to whole animal, but also say from phytoplankton to zooplankton to every species of fish to the little insects and critters that live in the sediment. We want to understand how it impacts ecosystem processing and function. And also, eventually, We want to use it to understand recovery, natural attenuation, and test some remediation strategies. It really is an opportunity to ask all the questions the government is trying to understand, really to understand how much we need to mitigate and how we need to mitigate. We have a very large team because if you are going to pollute a lake, you better ask every question under the sun, because this lake is taking one for the team and you really want to make sure you squeeze everything you can from it. So we have a big team, lots of different professors. You likely recognize some from the region, some are from the United States. And we have a project coordinator, Desiree Langenfeld, who did her master's as part of this project in the beginning and is now coordinating our whole ecosystem study. So for our whole lake study, we are doing what's called a BACI design, or a before after control impact. We have two lakes. Lake 378 is our experimental lake, and Lake 373 is our control lake, or our reference lake, which we never add plastic to. We study both of these lakes before we add any impact or pollution to Lake 378. We get to know its water chemistry. We get to know the every critter that lives in it, its population size, how healthy they are, etc. We do this for a few years. Then we add plastic to a few years, only to our experimental lake, and we ask all the same questions. And then we will stop adding plastic, and we will basically continue to do it again to understand recovery. So it's kind of actually like before, during, after, in our case, or before, after, after. But we're using this basically to understand, are any of the impacts we see happening over time, not because of plastic, or is it specifically because of the plastic? So that reference lake helps us understand if what we're seeing is from something else. We had a lot of discussion about the types of microplastics to add to these lakes, given that there's a lot of types of microplastics out there, and we want to make sure we do our study to be the most relevant to policy. So we chose microplastics that varied in size from 20 to 1400 micron, the whole size range. We chose fragments. They're among the most common in nature. And we chose 3 consumer plastics that are in really common items and that have different buoyancies. So low density polyethylene is positively buoyant. It floats. It's the yellow here. We did these bright colors so we could trace them. It's usually used to make buoys, kayaks, also bottles, and other types like grocery bags, etc. Polystyrene here is neutrally buoyant. This is not polystyrene foam. This would be like the polystyrene used to make solo cups, CDs, etc., CD cases. And then PET is a dense, it sinks quite quickly polymer, and it is used to make water bottles and usually detergent bottles. It's among the most recyclable plastic out there and quite commonly used in the universe. Then we had to figure out how to add plastic to the lake to best simulate contamination in the natural environment, and also not to add it in such a way that it would affect our questions. So for example, it's really common that microplastic enters a lake through like a pipe from a source. So for example, wastewater treatment plant effluent or stormwater, storm drains. But we didn't want to add it in particular places, because then those places might be more likely to have an impact than others. And then an effect we're seeing is based on our experimental design. So we decided that we would simulate stormwater input in terms of deciding how much we would add, but we decided we would add it evenly to the whole lake, basically running. We would drive a boat. We would put our microplastics in these buckets. This pipe would draw in water from the lake, and the water would go into our buckets, and it would mix with those cement mixers on top. Then the mixture would go out this pipe and go out the back of the boat where the motor is running, about a half a meter deep. Then we drive the boat along these transects. We had a boat driving these transects and a boat going around the perimeter to add plastics basically homogeneously to the lake. And I do this because you'll see as we add it, we do sort of get these stripes with the plastic. But here's just a video of us adding plastics to the lake. Our annual loading amount is 330 billion microplastics. We got there based on the amount that's been observed in stormwater in other studies, multiplied by the amount of runoff that enters our lake every year. But we decided to only add during the ice-free season, which makes sense. That is much more logistically feasible. And so we split up this amount by 12 biweekly additions. So every 2 weeks, we do these additions, adding about 77 kilograms of plastic per addition, equivalent to about a cup of plastic in an Olympic-sized swimming pool. And we do this for 3 years. This amount also would allow us to achieve concentrations in the water column that are equivalent to what we see in the Great Lakes, as well as amounts on the bottom that are equivalent to the Great Lakes. It's really important at ELA that you're not over polluting a lake, that you're choosing concentrations that would be seen in nature so that you can really answer questions about concentrations that would be seen in nature so you can inform policies today and not necessarily just projecting out into the future. So what I'm going to do today is give you a sneak peek on some of our results, then tell you how we're using this lake to test monitoring strategies that have been proposed under our IJC work group to sort of tee up Karen, Rebecca, and Eden for next week. And then if I have time, I'll talk about solutions to this issue. So for a sneak peek, I'm going to tell you a little bit about what we're seeing in terms of fate and a tiny bit about what we're seeing in terms of effects. So what I'm going to show you first is what we are seeing in terms of where the microplastic is going in the lake in the first 13 days, meaning after the first addition of that 1 cup to an Olympic-sized swimming pool added homogeneously to the lake after 13 days, which means before the second addition. So I'm showing you basically where did the plastic that we first added go within 2 weeks. The work is led by everybody on our team, but a lot of the work for this part has been done by myself, Matthew Hoffman, and PhD student Francis Nwama, who gets the most credit for helping sample and quantify and characterize all those microplastics with a huge team of undergraduate students. We have sampled 3 matrices or compartments in the lake for this assessment, and that includes the water column, basically taking a stratified sample from top to bottom at depth, and in the littoral zone, the shallow part of the lake, taking a depth integrated, so just taking the tube up and down and sampling the water at 6 stations and sampling at 1 day, 3 day, 7 day, and 13 day. We also have sediment traps, which are basically cups at the bottom. And as the plastic falls and sinks, it falls into it, using them as a measurement for how much is sinking down. And then us being able to extrapolate that across the bottom to understand how much has sunk and is on the bottom. And then sampling the shoreline or the beaches to understand how much is washing up. We sampled the sediment traps at the same time as the water column. And for shoreline, we're just missing that 3-day sample. We tried to sample across the lake to be able to capture that variability. So the data I'm going to show you is basically the average of these samples extrapolated across the entire lake. So of all the plastic we added, how much is on the bottom, how much is beaching, how much is in the water column, and how does that vary from 1 day to 7 day to 13 day? But before I show you that, I'll tell you we are only including the greater than 53 micron size fraction in this analysis. I would expect the smaller stuff to be in the water column longer, and we're in the process of measuring that today. It takes a bit of a different method and a different instrument. So the graphs I'm going to show you, I'm going to walk you through them. I'm going to show you for the total amount of microplastic and then for each polymer. So here what you're looking at, the total amount we added is this number of microplastics that's with this dotted line. Ignore the gray for now. The brown is how much is in the sediment. The blue is how much is in the water column. And the green is how much is in the shoreline. You can see at 1 day, the amount that's in each of these 3 matrices. And you can see how that varies at 7 day, where you have much less in the water column and much more on the shoreline and quite a bit in the sediment. This gray is how much we predict would be missing because we're missing that smaller size fraction if all of it was in these 3 matrices. So basically, this is the total amount of the smaller size fraction we added to the lake. And the outflow, you can't see because the amount leaving the lake is so small that it's negligible. But we also predicted that. So basically here you can see that we don't quite close the mass balance. We're at about 80%. Not including the smaller size fraction, including just the beaches, the water column, and the bottom. And what you can also see is that we add plastic to the water and it leaves very quickly. This is not quite the same story by polymer. Remember, the PE floats, the PS is neutrally buoyant, and the PET sinks. The PET, we accounted for basically all of it by looking at the bottom. For the polyethylene, we account for almost 80% of it on the shoreline. Most of it is floating and washing up on the beaches. And the polystyrene, we're missing a lot. We see some of it in the water column here, quite a bit of it sinking, but we're missing about 50% of this material. So the question is, where is the rest of the plastic? And we actually think we know. We think it's in matrices we have not looked at yet. So this is a nice visual that Madeline Milne made, basically looking at the amount in the different matrices at 1 day, 7 day, and 13 day. And you can see by 13 day, 23% is on the shoreline, only 4% is in the water column, and more than half is on the bottom. But we know some of it is escaping via the atmosphere, very small amount. Some of it's moving through the food web. Also, I think a pretty small amount relatively. I think most of it is right here in that shallow littoral zone, which we are looking at quite closely now, but weren't in those first 13 days, in the biofilm on the rocks, basically in the biofilm stuck to the macrophytes, floating on the surface water right here, and in those sediments. And we'll have that data soon. So basically, in conclusion, what we've learned is microplastics, though they are a very persistent compound, they last almost forever. They're very pseudo-persistent in the water column, meaning they leave quickly. The buoyant plastics wash up on shorelines. The dense plastics sink almost right away. We think the rest is likely, a lot of it is in this shallow littoral shoreline here. And this should inform what we understand about fate, but also exposure and risk, and how we decide where to monitor. And so over time, we will basically continue to assess this, and we'll do this across those more compartments. Now I'll show you a tiny bit about what we know about the impacts. So first of all, I'll show you that the animals are interacting with it. They are eating it. It's quite a small amount compared to what we add. But here is black-nosed shiner, pearl dace, spot-tail shiner, and yellow perch. And you can see microplastics in the stomach, in the livers, and in those fillets in the muscles. So just like in Lake Ontario, these fish are exposed to the microplastic and eating it, and it is going into these different tissues. This is the amount that we see in the zooplankton. This is before we added plastic. Good thing we're not seeing any at first. It's a good sign. After we add, you can see the amount in these different taxa of zooplankton. And after 7 day and 13 day, you can see those amounts decrease, just like they did in the water column. So this really begs the question, what's happening to these animals? And today I'm only going to show you a very early look at the phytoplankton and the zooplankton, the base of the food web. This is again work led by Madeline, who I highlighted in the Lake Ontario study earlier. Here you see the biomass, or the amount of phytoplankton in our experimental lake over the years. And here you can see that the concentrations are or the amount is pretty consistent. Until we add plastic, you see this drop, but it seems to bounce back by the second year. So are we seeing an effect? I don't know. Maybe it bounced back because perhaps the zooplankton are affected in year 2, which releases. So if you have a decrease here, you would imagine that the zooplankton might have been doing well here, and then they maybe decrease this population, and maybe then because of this decrease in population, the zooplankton later may decline because they have less food, and maybe this released the phytoplankton. We don't know. But in the zooplankton, we see a pretty steady amount, including in the first year of addition, and we do see this decrease in 2024. But I don't yet have results to show you for 2025, so this is very preliminary, and these patterns could go away once we have all the data. In addition to zooplankton, we looked at one of their predators, Chaobarus, which are kind of like a zooplankton, but they have a bit more control to swim. And these are midges that eventually come out of the water. And here you can see that their population isn't really affected over the years. It looks pretty similar. But here you can see the size of the Chaobarus in Lake 378 pre and post addition. And here you can see the size of the mysis in our reference lake, which are a similar species, not exactly the same. Here you can see a significant decrease in their size. Maybe they're getting less of their zooplankton diet in this year, whereas here you don't see a significant difference in the mysis. So it looks like preliminarily we are seeing impacts to the lower levels of the food chain, but it is still a little early to say with confidence. So basically what I showed you here is that microplastics are moving through the food web. They are translocating to other tissues. It seems like they might be affecting lower trophic levels, the primary productivity, zooplankton abundance, and growth, but it is preliminary, and we are in the process of looking at all of our data, and hopefully some of those impacts we start to understand very soon. So now I'm going to use the rest of my time to talk to you about how we're basically using this study to inform some of the work we've been doing in the IJC work group that Karen and Rebecca and Eden will talk about next week, and a little bit about solutions. So what Karen and Rebecca and Eden will talk about next week is an IJC work group on microplastic that developed monitoring protocols, strategies, risk assessment frameworks for microplastics that the governments can use to basically better understand the problem and motivate solutions. They're going to tell you all about it, but we tested one of these monitoring strategies in our lake. So we basically used our lake to understand how the methods we're proposing, how feasible they are. We propose sampling with a pump. So if you're ever sampling microplastics, we have an SOP that we could give you today that basically shows you how to sample with a pump. So taking water through sieves on a boat, as well as sampling with a manta. And people, of course, have questions about how might the results compare between these two methods. So here, this manta trawl has a 53 micron mesh, which is the same as our bottom sieve. So we compared them side by side at our lake at ELA. We also tested different types of pumps because some of them are cheaper to buy. So we ran 20 liters through each type 3 times and compared the output to see how the pumps compare to each other. We also were interested in the best size tubing. Should we get a bigger tube to capture all the plastics, or can we get a little bit smaller, which is more effective? Does it matter? You see people using all kinds of tube sizes across the literature, and we wanted to be able to recommend a size. And then we also wanted to help people understand how much volume do you need to sample in order to get a representative amount. So to look at the different pumps and the manta trawl, we sampled the same amount, although with manta, we sampled a little bit more because it's hard to get 20 liters. And here you can see we see no significant difference with any size fraction or in total across instruments. So you can buy whatever pump you want, which is great, or you could use a manta if you have one. We did not see differences in concentration by tube size, but we do recommend that the tube size is at least 3 times larger than the largest size of plastic you're trying to find. So if 1 millimeter is your top size, you should have at least a 3 millimeter tube. Volume is incredibly important. We recommend that the first time you go out and sample, you take at least 3 and ideally 5 concentrations, and you basically plot them side by side. Here you're seeing them plotted as the concentration, and here is the volume of the plastic of the water we took. Here you're seeing it plotted as the total microplastic count. The total microplastic count should be linear when you're getting the right amount, and the concentration should be the same when you're getting the right amount meaning a representative volume. Here you can see 10 is not enough. We get this random concentration that's much higher than what we would expect, which is typical. You tend to get higher concentrations when you undersample. But 20 liters and above is pretty consistent, and that's the sweet spot. So if you go out and you sample a few different concentrations, you can find the lowest concentration you can sample that will be representative, and lowest because it saves you time. You also want to look at the concentrations in your blank and try to make sure you're much below that. But essentially, our method was great. It was efficient. It was easy. And hey, it's not that expensive because you are welcome to use a gear pump, which is $100 to purchase. If anyone in this room would love to see those methods, I will very happily share them with you, and I'm happy to provide feedback and guidance on how to do that. So basically, to end with the pELAstic project, we added our final amount of plastic this year. We're done adding plastic. We're done polluting the lake, which is great. We are in the process of analyzing and synthesizing all that data to really better understand fate and effects. We are beginning this summer to study natural attenuation and trial some remediation strategy, and we're pretty excited about that, really getting into solutions. It's 2:43, so I want to be mindful of time. I'm going to spend somewhere between less than 5 minutes on solutions, and then I'm going to cut it. So if I cut through all my slides just to finish, I apologize, but I really want to have time for discussion. So I'm going to transition now to talk about what are the solutions of this problem. There is no one size fits all strategy to the issue of plastic pollution. It is a complex problem, and usually 3 solutions are kind of touted as the be all end all, and that is plastic waste reduction, which includes banning items, but also a circular economy. It is also waste management, actually picking up the waste and managing it in a place, even if it's an engineered landfill, that keeps it out of the environment. And the third is cleanup, which is good because it gets the plastic out of the environment, and it also monitors. We don't want to need cleanup. We would love to work our way out of cleanup with these other two solutions, but until we're there, we need all three. Right now, I think solutions are trying to happen at every level of government. The United Nations is trying to create the first ever plastics treaty. This keeps failing. They're trying to meet again this December. They were supposed to have an agreement 2 meetings ago, and this will be the third meeting where they were supposed to have an agreement. So we'll see what happens. But federally, our country is doing a lot. And I've been working on this issue for a very long time. I've been to UN meetings. I used to live in the United States. I am aware of what other countries are doing. And I can honestly say that I think we have some of the most comprehensive policy on this in the world. It's just that you're not seeing a lot of it happen quickly right now because there is a lawsuit. The federal government basically listed plastic as toxic under SEPA to allow it to ban the single-use plastic items you might be familiar with, but also to increase and improve recycling as well as producer pays principles through end-of-life responsibility or EPR. But this was struck down in the courts because industry sued the government, and right now there's an appeal, and depending on what happens, this policy may or may not continue. If it gets struck down, maybe different policies will turn on being led by federal government in other ways. This is all to say that I do think that we need this. I think we need federal policy, but I think for plastic pollution, local solutions are also really critical. Even with these federal policies, even with those 3 buckets I mentioned before, there will always continue to be some leakage of plastic going into the environment. And these things can be monitored and managed at the really local scale to help every jurisdiction understand what we can do locally to reduce our plastic pollution problem. So I just want to introduce you quickly to the U of T trash team and invite you to engage with us in any way, shape, or form that you're interested. I co-founded this organization with a colleague, Susan Debretzeni, in 2017, with a common goal to increase waste literacy in our community while reducing plastic pollution in our ecosystems. And we're empowered by youth, by students across the university and across Toronto. We have 3 work streams, which includes community outreach, which includes cleanup, education. We're in grade 5 classrooms across the city. We have lesson plans that you can beg, borrow, and steal for your own region. And we do a lot of solutions-based research. And that includes trash trapping, monitoring, but it also includes pollution prevention. And I'll talk a little bit about that. But the place I want to focus is we have created within our city a Toronto Inner Harbor floatable strategy where we brought together stakeholders in our city that would be the most relevant to this topic of plastic pollution in our watershed to work together on outreach, prevention, and cleanup/monitoring. Together, we have trash traps on our waterfront. We use these to clean up. We also use these for monitoring. Every summer, we remove more than 600 kilograms of plastic from our environment. We count up how much of it is macro and microplastics, but most importantly, we use it to understand the sources of plastic coming into the city of Toronto. You can see styrofoam is one of our major items. So there's cigarette butts, and this year, a lot of bottle caps. We also have a lot of small foam bits from construction and pellets from industry upstream that are making plastic. We use this data to inform these pollution prevention projects where we basically have students lead projects to better understand the sources of the item. So basically we find something in the environment, we better understand the sources and how it gets into the environment. We determine feasible solutions and we pilot them with the city to measure their effectiveness. And then we share those findings with stakeholders to inform solutions at scale. So for example, our first project, microfibers were among the most common item in our sample. We understood through our research that washing machines were a very large source. We trialed filters on the washing machines, and there is a bill right now in Ontario to put filters on washing machine to reduce microfibers. We are currently moving one of our pollution prevention projects to the city of Winnipeg to get reusable foodware at the Forks and other restaurants in the city. And finally, we would invite you to be part of our International Trash Trap Network, where we saw really great value in our cleaning and trapping for monitoring, that we created a global network, which includes resources for how you would do your own cleanups and trash trapping and education and outreach. It has an app to basically load your data into a globally accessible database. You can look at this data anytime by just creating a login. And then you can be part of our global impact. We are pretty new. We're about 4 years old. We are across 4 countries. We have lots of different types of trash traps, many subscribers, and together we remove hundreds of thousands of kilograms of plastic pollution from the environment every year. And we would love to have you contribute to our network and work with us somehow. So thank you so much. And I think I have 11 minutes for questions. Thanks, everyone.
Chelsea, thanks so much. That was fascinating. And I feel like I've learned a lot. I really appreciate how you walked us through some of the science of the project. I also really appreciate how many folks are involved with this type of science and research and monitoring. It really shows how much collaboration in ecosystem management and research is valued and important. Um, with that, I think I'll open the floor for if anyone has any questions or wants to have a bit of a discussion. You should be able to add your question in the Q&A box. Or raise your hand and then I can unmute you. Um, so see, Brady just said, thank you for your awesome presentation.
Thank you.
Does anyone have any questions or? We have a raised hand by Todd, so I'll allow you to talk, Todd.
Can you hear me now, Megan?
Yes.
Yep.
Good. Thank you, Chelsea. I'm sorry I wasn't able to tag into the webinar until a little bit late, but I did have a question related to the slide you showed in terms of effects on Cabris and mycin. And I appreciate there's no significant difference there, but there's just a hint preliminarily that there might be something going on. They're both vertical migrators, right? Any speculation as to what might possibly be going on there if there were a real difference?
Yeah, I think that so the mechanism of impact that we best understand for microplastic is a food dilution effect, meaning an animal eating plastic, filling its gut and getting less food and leading to changes in growth and eventually potentially reproduction. So it could be, we also know, and I didn't show data on this, that the Cabris are eating microplastics. So it could be that the Cabris are having a direct effect from the microplastics that they eat. But if we truly are seeing impacts to the phytoplankton and the zooplankton, I don't know if you saw the couple slides before, it could be that their food source is changing. We did look at, so before we started the whole lake experiment, we were doing these mesocosm experiments, and a postdoc in my lab, Garth Covington, was looking at the fatty acid composition of zooplankton and fish. And he found real changes in the fatty acid composition of the zooplankton, suggesting changes to the nutritional value at the lower levels of the food web. And I believe at the phytoplankton level as well, or the primary productivity. So what we could be seeing is food dilution affecting the nutritional value of every level of the food chain, or if it is truly shortening the food chain by changing the amounts of the zooplankton. So those are my like hand waving ideas as we really try to figure it out as we dig deeper into the analyses.
I appreciate that. That's good to hear, and I look forward to hearing more from your studies.
Thanks, Todd.
We have a question from Johan. I'll allow you to unmute if you want to ask it, Johan.
Hello there.
Hi, Johan.
Do you hear me?
Yes.
Yeah. Yeah, so I was just wondering if either you or anyone else on the call have a sense of like the most common types of microplastic specifically in the Rainy Lake of the Woods system. Like I understand our watershed is quite different than Lake Ontario. Yeah, like from those that we do know exist around here, any ideas of where they coming from? Because that would most likely give us an idea of how to prevent them from leaching into our waters.
Yeah, I would say that I personally don't know. And if anyone does, please feel free to answer this in the chat. But what I would say is that a great way to figure this out, if you are interested, is through running cleanups on your, like in your beaches, or getting trash trap devices. Some of them are really inexpensive, like booms, for example, can be put in places. We also use a lot of skimming to clean our watershed. So a lot of the debris will accumulate in boats, like slips for boats. So you can see whether you have hotspots of plastic pollution in your watershed, and then you can look at what it is you're collecting to get at the sources. And we have methods for this. We can absolutely send you materials. On the microplastic side, that's where you'd want to collaborate, I think, with someone who can run those analyses in the labs, because you don't need super fancy equipment, but you do need microscopy and usually spectroscopy. So like a Raman or an FTIR to confirm that it is microplastic. But I would say for the large plastic items, cleanups are a great way to increase awareness and a really good way to know the sources coming into your system.
Thanks, Chelsea. We have a few more questions. One from Stephen. I'll allow you to unmute.
Hi there, Chelsea.
Hi, Stephen.
I'm curious what results you were most surprised by in this experiment. Did anything stand out as like, well, that was unexpected?
I would say that I think one of the things that surprises us the most, even though in retrospect it shouldn't, is just how little of the plastic is in the water. If we think about, our modeling and where we would predict the plastic would go, we would have predicted that most of it would sink. I'm not sure we predicted the stuff on the surface would wash up on the beaches like it has, although in retrospect, reading the literature, we should have. But I think the reason it struck us as so surprising is because when you look at the literature, most people are sampling microplastics in the water. That's where they're monitoring, and they're consistently finding consistent concentrations in nature. But we know that once we add plastic, the amount we add doesn't stay there very long. So what that really suggests is that constant amount we see in the water column is due to constant input. The good news being, if we turn off the tap of the plastic pollution, the amount in the water column will attenuate fairly rapidly. And then we start to get the plastic on the beaches and at the bottom, which is much easier for remediation. But I would say really what's more important is eventually that will bury. And so what I really want to know now is how long does it take for it to bury to a depth where it's no longer biologically active? And I don't know the answer yet, but I would say that it really surprised us that we got to that question, that finding and that question so quickly. And now I'm so interested in the sediment and how it's moving through the sediment.
Yeah, that is interesting. Very cool. Thank you.
Thanks. For sake of time, we'll take one more. There's someone's hand up. Chris Hay, I'll allow you to talk.
Okay, great. Thank you. I'm really excited about the actions you shared about the trash team and the trash trapping programs. I definitely want to look those up. I guess I was curious, just from your perspective as like a plastic researcher that's been doing this for a long time, what are things that you do or that we should do to tackle the plastic problem, I guess? I'm thinking like on a personal level, like reusable take-home containers, cutlery. I've stopped eating seafood. I heard that fishing gear is a big problem. Or like even like writing to MPs about the federal policy, or like even at lower government levels. I don't know if there's policies right now or things that we should be pushing for. So just anything about that, I guess I'd be interested to hear what you have to say.
Yeah, great. Hi, Chris. I would say that on a personal level, so pre-policymaker, and then I'll get to that. I think one of the most important things we can do is increase our own waste literacy, meaning our knowledge about this issue, and then try to make behavioral changes that are relevant to solution. So it is really hard to change the convenient lifestyle we live in. I am guilty, just as guilty as anyone, of sometimes going to a coffee shop and getting a single-use cup because I forgot my mug, right? Or getting a plastic bag because I forgot my bag. But really trying to remember to go out of our way to reuse. And then trying to support those programs that are trying to make it easier on us. For example, there's companies that put reusable foodware in restaurants, and they make it so that it's really easy to borrow the cup and return the cup. So I think there's a lot of really great startups that are trying to make this easy. For example, one of the grocery stores in my neighborhood has they call it a bag library. So if you forget your bag, you can take a bag, and then the next time you bring back a bag or bring back another bag. But also, in addition, when we do have to buy things in single-use plastics, which we do often, if you're buying yogurt, if you're buying, most of the food we buy at the grocery store, try to buy the stuff that's in the packaging that can truly be recycled or does not go in the landfill. So for example, in the city of Toronto, I know that I can recycle a white yogurt container, but I know that I can't recycle a black one. And so I try to make sure that I get the containers that can go in my recycle bin and have a better chance of being recycled. On the policymaker front, we have a voice and they want to hear from us. And I think that depending on what happens to the lawsuit, some of the policies in place might get struck down. And so I think it's really important that we write to our federal MPs right now and tell them that it's important to us that we continue the policies we've started through other means, if necessary. For example, like having a private member's bill as opposed to the regulation. And then also a lot of, I think what we can do is at the city and the provincial level. So also writing, depending on where you are, your MPP or your MLA, and also your city councilors. So I do think that they do want to hear from us. And we have my students reach out all the time to politicians and they're meeting with them. And it's really fantastic being able to feel like you have that power in your community.
That's awesome. Thank you.
Mm-hmm.
Okay, so I think our time is up. Thank you so much, Chelsea, for joining us for such an engaging and insightful presentation, and sharing all of your time and expertise with us. It's really been fascinating to hear about the project. Hopefully we can have you back over the years to hear about the results. I'm really curious about the remediation efforts and what that looks like. And a big thank you to everyone who's joined us this afternoon and participated in questions and discussions. Just a reminder, this was recorded and it will be available on our website. You can also find recordings of our previous webinars from this series as well as previous years. You'll also be able to register for upcoming webinars in the series. As Chelsea mentioned, our next webinar is next week on Tuesday, November 4th. This one will actually be at 12 p.m. Central Time. And it will be on microplastics and freshwater health, specifically talking about lessons from the Great Lakes with Dr. Karen Kidd, Dr. Eden Hadley, and Dr. Rebecca Rooney. With that, thanks again, everyone. Take care and hopefully see you at our next session.
Thank you so much.
Thanks. Bye, all.